This method can help to answer key questions in the immunotherapy field about the assessment of immune cell cytotoxicity against 3D structured tumor cells. The main advantages of this method are that it is straightforward and it combines advanced imaging technology and a sensitive immunological assay.
Begin by washing the colorectal cancer cell line of interest with 5 milliliters of PBS and removing the cells from the flask bottom with 0.5 milliliters of trypsin for 5 minutes at 37 degrees Celsius. After confirming detachment under a microscope, neutralize the cell dissociation enzyme with 10 milliliters of complete medium and collect the cells by centrifugation. Resuspend the pellet in 5 milliliters of fresh complete medium for counting, and resuspend the cells at a 1.5 times 10 to the third cells per milliliter concentration. Then seed 200 microliters of cells into each well of a 96 well round bottom plate, and place the plate in an automated imaging apparatus inside a 37 degrees Celsius incubator with 5% CO2 and 95% humidity. Log into the acquisition software of the apparatus and select Schedule to Acquire, Launch Add Vessel, Scan on Schedule, and Create Vessel New. Next, select Scan Type Spheroid and select the appropriate bright field and fluorescence channels of interest. Set the magnification to 10 times and select the plate model and its position within the imaging apparatus drawer.